No, because they're not comparing your DNA to actual people. What they're doing is looking at your genotype and trying to figure out how often that pattern matches the pre-labeled populations that they are using in their calculator. If you were to look at your raw DNA file, you'd have a whole bunch of numbers on the side with this RS plus a number after that, and then with two different letters. So essentially, the RS IDs are the labels that we give to the different SNP positions or your DNA, and then the letters are the alleles. Each SNP position, you have these two letters, and that's what makes up your genotype. In this particular example, you could see how if we were just comparing a person's raw DNA to the presence of that genotype in the population, they would more closely match the Chinese group, because at 3 of the different SNP positions, they have exactly the same genotype that is present in the Chinese population. And they only overlap with the Yoruba sample population at 1 position. Why we even care about this in the first place is because the idea is that if you share these SNP positions, or you have an allelic similarity to a reference population, chances are you had a common ancestor with that population at some point. What the calculators do is you're never comparing directly your raw DNA to just what's in the population. There's always this level of frequencies that are added. So essentially, the researchers go in and say, yep, it's here, because most alleles, most genotypes are present all over the world. They're saying, how often do we see it in this area of the world? So that is dependent on them going to a place based on our current maps, our current boundaries, our current knowledge of people, and saying, this is the Yoruba group, and in Yoruba, we see this frequency, this percentage of the time. In China, with the Han Chinese group, we see this genotype, this percentage of the time. So again, it doesn't mean that one group has it and one group doesn't have it, although that can be possible. Like for example, the genotype GA that I just made up here, it's not present at all in the Chinese group, and it is present in the Yoruba group, although it is rare. Only 23% of the population would have it, meaning that close to 80% of the people in that sample do not have that genotype. And then you can see at the bottom, CC, where nobody has that. Nobody in the people that they've sampled. Again, it's not like they're going out and testing every single person available. They are developing a panel based on people who choose to be in the study. Okay, so then for every genotype at every SNP position, the algorithm, the calculator is checking to see, 1, is it present in the reference population, and 2, at what frequency? When something is more frequent in a population, it weights the score towards that group. So for example, if you're looking at the first SNP position, the 101, and you're looking at the genotype there, it's present in both the Chinese population and the Yoruban population. But if you go down to the frequency panel, it's seen more often in the Yoruban population at 81%. So the calculator says, we're going to give this over to Yoruba because it's more often, it occurs more often there. And essentially this is done thousands of times, where it's going through each different position and it's looking at if you have that genotype, if that genotype matches the reference population, and then how often does it occur. Wherever it occurs more frequently, the weight is given there. Then you get things like the CC, where it's not present in either population. So if you look at the middle of the frequency, it's not actually present in the Chinese population or the Yoruba population. However, if you look down there, we do know that at the allelic level, the C does show up in the Yoruba population. So because nothing can be absent in these calculators, everything has to be assigned to a group, the weight is given to Yoruba, even though the genotype of person X doesn't match either one of them. It literally, none of these groups have CC, but it's picking the closest fit that kind of work. Person's individual DNA actually is more similar to the Chinese group based on the calculator and how the calculator is weighting using frequencies. It's assigning it to the Yoruban group, even though there really isn't much genetic similarity between person X and that sample population. So it doesn't mean that that person is actually related to the Yoruban group. It just means that those letters happen to appear more frequently in that particular population group panel. So the calculator is never reading who you are. It's literally just comparing you to frequency tables. The DNA calculation isn't about ancestry, about family. It is literally just frequency matching. And because our society assumes that we know why certain groups match, that's where we get this whole idea that if you closely map a population, then you're descended from them. But it doesn't explain other possible reasons that you and another group could be similarly matching. It also doesn't explain why certain genotypes show up more frequently in certain areas of the world or with certain populations beyond the agreed upon assumptions.